If it's loose, is that?
Yeah, I want to, yeah, anything that could break off, or if there's something loose in this little bumpy environment would be excellent.
Okay.
So maybe that ledge that's like casting a shadow just above the lasers.
Okay. See?
I'm seeing in the chat that this might not all be basalt, so it could be breakable.
This piece right here doesn't interest you?
It looks easier, huh?
Just saying.
If we don't rotate it too much, I can take a look at it. If we can zoom in once he's ready.
Okay. Zoom in.
Possible. A little more of a rotate. It actually kind of looks like it could be the phosphorite we've been cutting. Can you go pan up just a little bit? Could have a crust on it. We could take it. Is it too big?
No, we need rocks, actually.
We need weight.
The more the merrier. So.
Okay, let's sample this one, and I'd love if you can nudge that other ledge that was above it.
Okay. Where's this going?
The shift, they're just getting on.
Probably starboard, because I think the colony experiments are still in the front. So.
Oh, right. So it's starboard no matter what.
Yeah.
Yes, that's true.
Want to follow me around?
Yeah.
We're looking for a rock spot. Or you can also choose it and
sample
let us
salvo.
know. Yeah, it looks like we should go for
Popping out.
the aft starboard bio box outboard.
Aft outboard.
Aft outboard.
Yes, please. Oh, that's a decent sized rock. Which is technically, that says 3 pink, which sometimes isn't true, but we'll go for that.
Wonder if it'll crack.
Is it too big?
No, I think I just got a bad angle on a dangle there.
Angle. What sample number is this?
Oh, it broke. Broken too. All right.
Oh, but it's in the box?
It's in the box.
NA12415.
155.
And this is going to be rock sample number 2.
Rock 2.
Caitlin, can you bring your mic just a little closer?
Aft. Outboard.
So you wanted something like off of right there? Where the
a little to the left. But the one that's casting a shadow. Yep, right there.
Right in there?
Yeah, if that broke off, I would be so happy.
Well, that's what we're shooting for.
I got one. Yeah.
Oh, it's a dirt clod.
Okay. So there's our answer, not basalt. And it's too crumbly to grab one, huh?
Well, depends on what size you want.
There's that,
zoom in.
Sorry, now can you pull up that target? I missed the
yes.
the last rock, thank you.
You want to zoom in some more? What do we think of this rock?
Is that the one that just fell?
It's the one that broke.
I think it's interesting, but we can't use it for bio if it's all fallen and broken.
There was a nice chunk with a brittle star on it on the wider view.
Okay, so moving on, zoom out.
You want us to poke around at other rocks here?
Yeah.
Bob can do that.
Yeah, I was looking at that one that's sticking out.
With
Right where your claw is, that little knob.
the
Yeah.
little star or something on it?
Yeah.
Might be, I gotta relocate.
Oh, okay. I just thought maybe it was easier to grab.
Well, it could be, I just have to relocate a little bit. No big
Another.
deal. You want it? Hey, Jake, can you go to the Auto XY page and see what the heading is set to?
Oh, why am I pressing the screen?
You got it dialed way down. Crazy low.
You want me to?
Yeah, turn that to
Oh, the
2.
step.
Make it 2.
Okay.
Point price is crazy. I don't like it.
Half a degree.
It'll take me 8 hours to turn.
It'll take you 720 clicks to turn around.
So are we thinking maybe that rock kind of in the mid-screen there, the
Yeah.
knob kind of sticking out?
Yeah, any of those little knobbies that are.
Not a great landing spot.
And for those watching, the reason I'm shooting for the ones that look a little bit more rounded and knobby in the upper left of the view, I'm hoping that that is a ferromanganese coating on the outside, that we can look at the microbiology on the ferromanganese minerals and look at the metal concentrations and that versus these lower rocks that are more angular and look to have less of a crest on them. I also like challenging Bob to try and grab random things for me.
Yeah.
One of my favorite activities.
Can we zoom in on that knobby thing? Zoom out a little bit.
Ooh.
Oh.
Is it working?
It's coming.
Oh.
It's okay. Not everything fell off, but a lot came off.
Wow, you guys are picky.
I mean,
Porch rock.
yeah, we could take it in another compartment if Lisa doesn't think it's good enough for quant bio. What do you think, Paul? Too many things fell off?
I just saw a tuna crab leave or squat lobster leave, but that's probably inevitable.
This is, yeah, I mean, this is heavy rock excavation trying to hang on to little bitty
So this crust looks brittle, so that's encouraging.
Yeah. Yeah, that more delicate, bumpy outer texture, but this seems to be a sedimentary rock, a lot of the.
This is going to be big.
Ask
Need
for
big.
rocks.
All right. Well, goodbye, rock.
Out.
There you go. Long gone?
That might be it.
It's okay to be a porch rock. Definitely not a bio-quant rock now.
Porch rock's okay too. Guess it's gone. Long gone.
What do you think, Greg? Do you think too much biology came off of it?
Was it attached?
It was attached to the, yeah, we just pulled it from the outcrop.
Yeah, I think she'd rather have a free rock because we haven't had attached rocks before.
I don't know if it matters as long as it looks like the biology stayed on it, right?
Have you had attached rocks before? Because she wants, you know, the underneath fauna as well as the above.
Mm.
Was it only attached in a small place?
I think just that one point there.
Yeah.
They're cool striations on it.
Let's take it.
Definitely want to keep it if you think this is
Yeah,
a
I definitely want
ferromanganese
to.
crust.
Definitely want to keep it. Do we think
We
enough
could
biology?
put it in a non-box box.
Do we
We're
think?
not trying to
Crevice
keep
of the boxes.
Do we think bio fell off of it to make it non-quant? Greg, did you notice?
I didn't see anything fall off it.
Yeah, okay. Box it.
Let's keep it as a
Bio.
quantitative bio box rock.
Beautiful. So if we could go for the starboard bio box, forward inboard.
Forward and board. That would be yellow if you have color vision.
Or number 2, if you like numbers.
2 yellow. Fingers crossed.
But I think it's also, we're now writing also inboard,
See
outboard.
what I'm doing there? We got the wall right there, so I don't want to.
Yeah, okay. As far as bubble cam can go. Forward inboard.
Forward inboard.
Yeah, out more.
Inboard, I believe.
Forward inboard, is that correct?
Yes, let's go for
Inboard is-
inboard if
inboard's
possible.
Oh, yeah.
Beautiful. We can see that yellow. And this is going to be sample NA124-156, rock number 3.
Copy.
Okay. Thank you for my 2 rocks. I will relinquish my shift now. You guys enjoy.
Go eat lunch.
I hope you find all the best for Gorgonian sponges.
We will try our best. All right, so let me introduce the second science watch. In the back row, we have Caitlin, myself, Paul Jensen, and Greg Rouse. Kylie and Caitlin are both graduate students, and Greg and I are both professors at Scripps Institution of Oceanography. In the front row, we have a slight deviation from our usual arrangement. We have Bob as the sub-pilot. More used to Jessica, but it's going to be fun to work with you, Bob. And we've got Jake piloting the Argus. And we have Rennie as navigator. And we have Jonathan as our expert video man who reminds us when we have our mics poorly positioned.
Okay, so from what I understood from the last watch, this was kind of the first major significant rocky outcrop that they were interested in. So I don't know if you want to interrogate it anymore before we start to move on towards the waypoints. You can see on HiPAC we've got slope map up. These lines are the general track of these ridges. They're not super steep ridges. And then we're headed to these intermediate waypoints, AIA and AIB, on our way to AJ, where we will supposedly deploy the other experiment. But anything you'd like on the way there, give a shout.
So where are we going?
Sounds great.
Would you like to look at this before we start moving, or keep onward?
I'm ready to move.
Okay.
Ready. Looks like, just to review, we've had one transect done so far.
Yeah, we haven't done a transect for a while. So we're over 100 meters shallower.
Mm-hmm.
Yeah, so maybe we start with a
Yeah.
transect.
Let's knock off a transect.
Okay.
That sounds great. This will be transect number 2.
Transect 2. So that'll get us right to this kind of steeper bit. Bob and Jake will kind of be cross-cutting contours, so we'll be ascending but moving along them as well.
What's the...
So not directly up. It looks like we should go, if we're on our way to AIB, 115.
115.
115.
120.
120.
120.
And then if we could possibly have a confirmation when we start our transect, that would be great. Thank you.
Sure. I'll confirm. So we'll just, once we get all set up at 120, we'll just wait for Argus to move once I call that in, and then we'll start.
Greg, what do you think? Does that look like that primnoid we collected yesterday?
Bridge nav.
I think we have had that, Paul.
Or is that a sponge?
Yeah,
We'll
it's
have
a
a
sponge.
ship move
Okay.
Yeah.
Oh yeah, sorry, it is definitely a
100
sponge.
meters, bearing 120, speed 0.3.
So these transects are another way to document the biodiversity along these rocky ledges. So we're trying to do 5 of these per dive at 100 meters each. And at some point, someone will review this video footage and count the different types of organisms that they see, their abundance and diversity. It's a non-collecting method of documenting what's here.
All right, we can call this the start of transect right here.
Thank you.
NA124, H1845.
You're just joining us. We're on the southern side of the 40-mile bank off of San Diego, so it's about 40 miles offshore of San
It's
Diego.
flatter over here.
At 870 meters currently.
I'm
Pretty
tilted up a little
sedimented.
bit high for flat land.
Oh, the camera? Okay, yeah.
All the way out.
So we're hoping to complete this transect without getting too enamored with something that we would like to collect. But if we do see something special,
Okay, a little more zoom. Just junk zoom.
There we go. Junk zoom.
Junk zoom.
Pass the junk.
So it'll be really interesting about this transect is we'll be able to compare this 100-meter video transect to all the other transects that we've had at all the different dive sites. So these could be at different depths, at different features, and then going back and doing the quantitative analysis of all the different species and what those different ecosystems look like will be a really good idea of the diversity and creatures we might be seeing compared across all the sites. So we try to aim to do 5 of these transects at each of our dive sites. So this is our second one today.
We're seeing lots of marine snow in the water column. The little guys that you see dashing around are zooplankton, maybe copepods.
And lots of sediment.
It's also a few burrows in the foreground and a thorny head rockfish in the lower right. Euphaucid elegantly swimming by.
I see your method, Bob. You adjust the joy gain. That adjusts it live rather than keep adding in the different XY speeds. Interesting. Noted.
I saw Jess do that yesterday.
Mm.
She was messing around with it.
So Bob, I don't know what happens on the first shift, but we tend to have contests on the second shift for who can spot the best prize. And one of the things we're particularly interested in are...
What's the...
Yeah,
what do you win?
What do you get? Yeah. That's the surprise.
We're all here. Ooh, here's a nice hagfish coming in.
Joy.
Yeah.
Joy of deep sea exploration, Bob. Same price as always.
You get your name and the acknowledgement of a publication.
Oh,
Really?
I'll take that.
But one of the things we're anxious to collect are some octocorals, especially some formerly known as gorgonians.
Oh,
Very cool tulip sponge.
I was going to say it looks like a tulip.
It really does look just like a tulip.
It's called hyalonema, otherwise known as a glass rope sponge
Glass ropes.
or a tulip sponge.
Beautiful.
And they often have a sea anemone sitting just under the filtering part. The last two we just saw didn't.
I think the one on the left just did, actually.
It did?
Cool.
A little white one, yeah.
Tiny white one?
Yeah, but there can be a big Leponema pom-pom anemone often sitting there, and it looks like a hybrid creature.
Oh.
the glass rods are several millimeters thick. Quite an amazing thing for a simple animal, we'll call a sponge, to secrete. And the way they wind around each other in a helix gives them a lot of stability in the current. Here you can see dead ones where the glass fibers are still coming out of the sediment. Who knows how long after the main body of the sponge died.
Yeah, I was just about to ask Greg, do we know how long those glass stalks will stick around for?
No, I don't, Caitlin. I don't know if anyone's tagged one and followed them. You need someone who comes back to the same place year after year to map and track these things. We do have a whale fall that Nautilus has visited called Rosebud, and when that whale fell, there were a lot of hyalinema around it, and several of them were killed by the whale fall, and we could review all the video footage over the years and actually see how many have continued. That's something we should do one day.
I know we were collecting a lot of samples. We're seeing a lot of interesting invertebrate assemblages on top of kind of sponge graveyard areas, so to say, kind of the skeletons left behind. So be curious if over time they're slowly broken down or continually provide some other type of habitat, even when they're perhaps dead and no longer growing.
Bridge nav. Add 20 meters, 120.
Ooh.
I can slow to 0.2 if you like, Bob, but it might not react as quick as just ascending.
Neat.
So Greg, has anyone ever figured out when there were those big squat lobster die-offs or tuna crab die-offs that washed up on the beach in San Diego? What, is that an oxygen thing?
Not really, because the tuna crabs can tolerate low oxygen pretty well. We found them in at a place where the oxygen minimum zone hit the seafloor off Costa Rica at about 450 meters. And all that was living there was a bacterial mat and millions and millions of tuna crabs, plus an occasional predatory mantis shrimp. And so that suggests they're really able to tolerate the low oxygen. And yet we've seen massive die-offs and events where they wash ashore in Southern California. So it wasn't really clear what killed them all. They often seem to wash up alive, and we took some and put them into our aquarium system, and the animals recovered. So I was almost wondering if they washed ashore just by an accident of occurrence, rather than dying. And so they were washed ashore while they were still alive, and for some reason they couldn't get away, or the water was possibly warm, too warm for them, and they're more of a problem in warm water than what they're used to in cooler water. But that said, they tend to expand northward from Baja and further south when the water warms off California. So It's a bit of a mystery the way their populations expand and contract. But what we're seeing are relatives of tuna crabs, another kind of squat lobster probably called Munida. We saw definitely tuna crabs yesterday at the Cortez Bank when we were much shallower. Tuna crabs have more white and red mottling, but we haven't had a real close-up of these squat lobsters today. We should try and get one if we can with a slope.
So do they spend part of their life cycle in the water column, or at least what do they feed in the water column, or are they always on the bottom?
Yeah, they can do both. They can swim, and that's where they get their common name. Tuna crabs is they're often up in the water column and being preyed on by pelagic fish like tuna. But they're probably, and they can feed in the plankton when there's a lot of stuff like marine snow, or they can perfectly happily live on the bottom. We've trawled them just near here, where they have completely filled an otter trawl with, I had a class estimate how many tuna crabs we caught on a 20-minute trawl on the bottom, and it was nearly 400,000 tuna crabs. Completely filled the cod end of our otter trawl.
All right, that's the end of transect right there.
Beautiful, thank you.
Thank you.
And the massive number of these tuna crabs on the seafloor must be having an effect on the ecology of the organisms living in the sediment, and they've been there now for a number of years, and it almost as if it's they're going to become a regular feature of the Southern California fauna rather than an episodic.
So that's a new observation to see them in
that
those
we're
kinds
seeing
of high numbers.
Yeah, and that we see them so consistently now, year after year.
Okay, so if you take a look at the slope map I have up here, you can see that we kind of just cross-cut a bit of that red bit, but on our way forward to AJ and then later AK, there's not really that much. So if you're interested in this steep slope, what you've been seeing here, we could try to kind of go along contour by going south on our way up over east to AJ, or we can just head straight to AJ if this isn't.
Contour for a while?
Yeah, I think so.
Okay. See if we can find some prizes.
Sure.
So what are we looking for, Paul?
So Bob, I'm gonna move 200, and we'll kind of be facing the wall, moving along contour-ish for a little bit. See what we can get.
Yeah, so one of the prizes we're seeking are a group of octocorals that, as Steve pointed out to us last night, are formerly known as gorgonians, not a proper taxonomic term any longer. But this is a group of octocorals that are well known for being rich in these molecules we call natural products, which I think we're going to be talking about a fair amount during our watch. So we're very interested in collecting some of these deep water species because they haven't been studied for this purpose. This is usually about the time that Kylie breaks into a joke.
I'll put my thinking cap on and try and come up with something.
Going by a pretty cool Venus flytrap anemone. The edge of the rock there.
Can we zoom in?
Zooming in.
cool.
Yeah.
Next to a sponge and next to a dead sponge.
Okay, zoom out. Anyway,
How about that?
junk zoom.
Look at that steep escarpment.
Yeah.
Mm.
Looks very different from those sediment plains we were just flying over.
Interesting texture on that rock.
It's a large solitary hydroid to the right of the lasers.
What's in the bottom left, that purple thing?
You zoom in.
Good question.
Is that an octopus?
Mm.
It could be. I thought I saw some big eyes.
Yeah, I
Lower
thought of some big eyes.
purple blob in
Back there.
the crack.
Right
Yeah,
Oh,
there.
down there.
little folded up
Oh.
octopus.
Wow, look at him.
Is the second purple one we saw?
Yeah.
I've seen a bunch
I
of
don't
orange
know.
ones, 3 orange ones.
Look at that cute little cephalopod.
Can I go in tight?
Oh.
Yep.
Oh, how cute.
Oh my goodness.
Look at his little googly eyes.
It's so cool to be seeing these amazing creatures in real time. They almost look fake, but
Oh.
there they are, the bottom of the ocean floor. Looking so cute.
Really cool colored octopus.
Like his bulgy little eyes.
Rennie, can you give me a stick lock?
Stick lock.
There's some big pupils to absorb all the light it can in the deep sea.
Nice. All tucked away.
I'm getting some great screen grabs, thank you.
Nice.
So we
That
good?
little guy.
Good on him?
I'd
Move it
say.
on. Zoom out.
Yeah.
See you, man. Or girl.
Zoom out.
Or both.
You come out on Argus as well.
Coming out.
Thank you.
So Steve tells us this is a bobtail squid.
Bobtail.
You know, there's a species of bobtail squid in Hawaii that has bioluminescent
I'm a
bacterial
bit ahead,
symbionts.
Bob, so I can pause.
Right.
Bridge nav.
That's so cool.
Hold position. Thank you.
Yeah, so these bobtail squid in Hawaii use these bacteria that generate light, and they maintain them in a special light organ under their eyes, or near their eyes at least. And they use those to recognize each other. They also have symbionts that generate, bacterial symbionts that generate light that help provide them with counter shading to avoid predation. It's a pretty amazing symbiosis. There's a couple of groups around the world that have been studying them and trying to understand how these squid basically have these monocultures of bacteria growing in these little pouches. They expel them every evening, and then they take up new ones and they grow inside these light organs, and when they reach high enough density, they emit light. It's a pretty fascinating symbiosis.
Want to zoom in?
Coming in.
It looks like the Pharea, right?
Oh, wow.
first sponge we collected on this trip.
Very cool zoom.
Okay. You can zoom out. Move on.
All right, I'll call in another one. We'll still go 200, 200 for another 30 meters. Bridge nav.
We, zoom in again.
Swiftia
How much? Oh, on this?
Or
or
Paragorgia?
bubblegum.
30
Paragorgia.
meters, bearing
Bubblegum coral?
200.
With its polyps out feeding,
Thank
it
you.
looks like.
Hmm.
So that's an example of one of the prizes. We've collected 2 samples so far of this animal. We may collect another, but I don't think we'll go for this one at this point.
Swiftia.
Is it Swiftia?
It's Swiftia.
Yeah, okay.
Swiftia.
Get more zoom?
Yeah. See that?
Swiftia.
Look at all those little polyps grabbing plankton, drifting past. Corals that live in the upper pelagic have a symbiotic phytoplankton called zooxanthellae, and the zooxanthellae turn sunlight into energy that the coral kind of steals.
Okay, zoom out.
Coming up.
But in the deep sea, there's no sunlight, so these gorgeous corals have adapted to be hunters and catch their food.
So we have a note in the science chat. Mark Legg, who has joined us, is saying to note the orthogonal fractures and joints that cut into the scarp. These could be conjugate fractures, some of these rocks that we're going over.
So question for Rene or the subcrew: does the direction the ship is heading affect you? Does it? Cause sometimes you're.
Oh, you mean the heading of the ship?
Yeah, because sometimes we're leading from the stern, sometimes we're moving sort of.
It mostly doesn't. The one thing that we have is we have a camera right there in the upper right. That's the wire cam.
Right.
And if we were moving backwards over that, over extended periods, we just want to make sure that that wire doesn't ever touch the back of the ship.
Sure.
But oftentimes there's enough kind of cable out that it doesn't quite, it's far away enough that we're okay to move backwards over it. And because it is a rigid steel cable with a heavy weight on the end.
So
But there are some times when we don't want to move.
Is there a preferred direction? Is the ship is moving forward or back or?
So the ship in dynamic positioning, it tends to, it prefers to be facing into whatever forces there are. That's kind of, it has the most capability to be facing into the wind and into the current and into the swell. Otherwise, we'll either get jostled around or the ship will fall off station. Just because you take a strong wind abeam of the ship, and it has
We zoom in.
a lot of trouble holding station.
Sure, that makes sense.
So they kind of optimize what's good for the ship, and then we move around kind of laterally from there.
Thank you.
Mm-hmm.
Crab looks like its claws seen better days.
Okay, zoom out.
Is that a dead shrimp or something on its back?
Can we zoom in on that little lobster?
Whoa.
Yeah,
What kind?
Whoa.
What are you?
that's a blind lobster.
Wow, I've never seen that.
Blind
Yeah.
lobster.
That's cool.
It's got these delicate little appendages up front with claws on it.
Ooh.
You can see
Oh
them
my goodness.
coming out.
Wow.
Jake, you ready for the ultimate slurp challenge here?
A little big for that. Wow.
Another adaptation we see in the deep sea is loss of eyes. Because why would you need eyes if there's no light?
You can zoom in.
Coming in.
Made his own dust cloud, though.
Wow.
I'm not sure if they really are blind. It's their common name.
Oh.
Is it related to a slipper lobster?
Yeah, indirectly.
Sort of.
In the same kind of ballpark. Yeah. Of Decapoda.
Are we looking at towards the head?
Yeah.
Very
Look at
cool.
him face
Yeah.
on, right? Looks like he's got eyes.
We're still moving, Rennie?
The ship is now stopped, but Argus
Okay.
is swinging a bit, so.
He's
looking-
digging in.
Yeah, he's digging
Yeah,
in.
burrowing back.
Yeah, they'll hide just below the sediment, dusted over, and just with their antennae out, waiting to pounce with those claws.
Yeah.
They're like
Do we
little
ever-
scalpels, those two claws they have.
Reminds.
Or you might start to get dragged there.
Long tweezers.
Hey, gotta move on. Move on.
Reminds me of those. I think it's incorrect. But pseudo scorpions, the
Yeah.
on land, the little guys
Yeah.
that.
Yeah.
With those really long.
Yeah.
Yeah.
Yeah, with no tail.
Yeah. A couple more. Like, yeah. Very cool.
Can we zoom in?
Urchin?
Yeah, that's the
I think
Ariosoma, the
that's sight
squish,
on, that's why it looks
Yeah.
weird.
squishy urchin.
I thought it was a sponge from far away with the spicules. Profile view.
So those blind lobsters are fairly closely related to true lobsters, the ones that people might know from the East Coast and Europe that they like to eat. Sometimes they're put together in the same group.
Something big just left that spot. Little cloud of dust.
All right, so I'm gonna kind of continue along contour, and we'll be going 155. Bridge nav. .50 meters, 155.
155.
I have to say, in comparison to many of the other sites, this is not as great an abundance of biology.
All right, I got my first joke. When should you be afraid of an invertebrate?
When you're feeling spineless.
Is that it?
Oh, I hope that was it.
When it's anemone,
Uh,
like an enemy.
oh, we got it.
Is that joining the list or anything?
I might get on the list. Yeah. Banned joke list. No. So far, just this tailless slope, not really any kind of outcrop walls, except for that one at the beginning. We are on kind of the upper part of this slopey region.
A little white blob is.
Where?
Crispin on the science chat suggesting this is the lobster we saw before. The blind lobster was polycheles, and could be Crispin, could be a polychelidae, it could be a typhlo chelidae. Didn't quite get a good enough look at it to say. Polychelidae is a more diverse group, so it's probably a better guess.
Kind of is that a dead sponge or a rock there to the left of the lasers?
Up ahead?
Looks like one of
That
those
one?
sponges. Yeah.
Dead
Yeah,
sponge.
dead sponge.
Yeah. I notice we're seeing some of those kind of brown, red feather duster worms. Kind of some dotted in the sediment. Seeing a couple of those. In the
Yeah, that's a feather duster worm that lives in a sort of a transparent, slimy tube in the seafloor. They're called Mixicula. They're quite a famous worm for physiologists in that they have very giant nerve cells in their ventral nerve cord, which meant in the early days of neurophysiology they were amenable to experiments before we developed the better instrumentation that we had today. So squid axons were one of the early tools, but also the nerve fibers of that particular worm, which has got a fantastic withdrawal mechanism when it feels threatened, and that's facilitated by having these giant nerve fibers. Luckily, we did catch one to document what it is, so Jessica slurped one up the other day, which I was happy about.
Oh, very cool. Were you able to get the tube as well, or did that stay in the
tube
sediment?
is literally mucus.
Oh, okay.
Pretty...
Oh, chair.
Huh.
Is that
It's a table.
a deck chair?
That?
Zoom in?
It's a nice anemone.
Very artfully poised anemone.
Guess we could mark that as some type of trash.
Trash, yeah.
Not sure I'd want to sit in a three-legged deck chair.
Neither did whoever threw it off the side.
Hey, zoom up.
Mark's chiming in on the science chat about that dead sponge that we saw, saying that could have been knocked over by density current or debris flow, which definitely looked big enough for, once it had died, something could have fallen over there, now covered a sediment, now a home for different types of creatures.
Too far back.
I don't know. Renee, can we zoom out so
Sure.
we can see what the topology is up there? Oh, it's a long, flat section above us.
Yeah, so right now I've just been kind of hanging out in this red bit, but whenever you want, we can jog east towards our HA.
It's a bit of a steep section straight west of us now.
Down here, yeah, so that's
Yeah.
downslope. Kind of.
Oh, I'm turned around.
Yeah,
Okay.
sorry, here, let me put on the, let me just do a quick,
That's downslope. Got it.
where are we? There we go. So that's kind of the bathy. East is generally upslope.
But the experiments were deployed, they were deployed at 1,000 meters.
They were deployed somewhere back in this.
Oh, I see. I was misreading it. AJ is where we're meant to deploy.
Yes,
the depth contour we're meant to deploy.
Yeah,
I thought that's the one where we had deployed. Got it. Okay, sorry, we can zoom in and we'll have a better assessment.
Sure. You like slope map back on?
Yes, thanks. Oh, no, let's just keep traversing, and then when we get to the section south of the deployment, we can head up.
Okay.
Yeah.
We'll kind of head around here.
Yeah, sounds good.
Something like that. Okay.
Trying to zoom in on that.
Bridge nav. 50 meters, bearing 170.
Okay, zoom out. Definitely going down the slope now.
Yeah, we're in a, you can see in Argus, we're in a bit of a pit, and we'll be coming back up.
That shrimp that we saw a while back had a really shiny looking eyeball. And another deep sea adaptation is to have something called a tapetum lucidum, which is kind of like a sheath behind the cornea in an eyeball. And what it does is it refracts light back out the eye through the cornea to make use of all of the light that's available in the deep sea. So a lot of deep sea creatures have this tapetum lucidum, and you'll see their eyeballs kind of like an iridescent shine. And also terrestrial animals have it too. Like when you take a picture of a cat, you see their eyeballs flare up in the flash. That's because of the tapetum lucidum. They've evolved to be able to see at night. Yeah, so cool.
Sorry, did you say zoom out?
Yeah.